Hi, I'm Zondo, and this is my partner Daphne. In our experiment, we wanted to see if temperature affected the radiance of luminol. Here were our purposes.
The first purpose of this experiment is to learn how temperature affects the amount of light produced in the luminol reaction. Our second purpose of this experiment is to understand how the light reaction reacts towards different temperatures.
Our hypothesis in this experiment was if the temperature of luminol is changed, then the amount of light produced will increase when luminol is in a heated temperature.
The independent variable in this experiment is the temperature, and the dependent variable is the intensity of the light. The control group is the luminol at room temperature, and the constant variables are the amount of luminol, the test tubes, the process used during the experiment, and the amount of water.
Some background information that we needed to know before experimenting was that the glow of luminol is called chemiluminescence. Luminol is a chemical that will emit light after reacting with sodium perborate in water, and this will later produce hydrogen peroxide. Hydrogen peroxide is an oxidizer which, in presence of a catalyst, will cause the luminol molecules to enter an excited state. The excited state gives out energy as light when it decays to the ground state. Something we found interesting about luminol is the fact that it can detect blood that has been diluted up to 10,000 times, which is why it's so important in investigating crime scenes. The materials that we used for this experiment were an instant luminol powder, 3 clear cups, a liquid measuring cup, a thermometer, Poland Spring water, plastic spoons, a phone camera, a stopwatch, gloves, safety glasses, and test tubes.
Our procedure was very simple. We only needed 3 bottles of Poland Spring water, and then we put 400 milliliters of water into 3 beakers. We heated a beaker to 60 degrees Celsius, and then we refrigerate one of them, and then we kept one at room temperature. We placed 3 tubes into the holder, and then we measured about 4 grams of luminol for all 3 tubes. We poured luminol into the tubes using a funnel. Then we measured about 23 grams of water into 3 tubes. We poured water in tubes into the luminol tube, and then we stirred water and the luminol mixture in the tube. We observed and we took pictures of the glow.
Some main things that we observed was that the hot water had the brightest glow, the quickest reaction time to the luminol, but something we found interesting was the fact that the glow had expired the quickest. So in this picture, we're still pouring in the luminol powder into the hot water, and here is immediately after pouring it in. Another thing that we observed was the way it looked while reacting. We noticed that it kind of looks like a flame arising while we added it. In this picture, it's most visible. You can see it kind of arising. After adding in the luminol powder to each different temperature water, we also noticed that each different temperature gave a different kind of glow. So in the cold water, it gave the weakest glow. In the warm water, it gave a glow brighter than the cold, but still less bright than the hot. And then the hot had the brightest glow. Lastly, we observed that even with the lights on, we could still see a yellowish glow, and we correlated that to kind of looking like oil.
The hypothesis for the luminol experiment is if the temperature of luminol is changed and the amount of light produced will increase when luminol is heated. The hypothesis was supported. For example, the warm and the heated temperature glowed the brightest compared to the cold and room temperature glow. Luminol activates with an oxidant, which usually contains hydrogen peroxide. Oh my gosh. Hydrogen ions in the water activates with the glow. And this is why the first experiment didn't go so well. As you can see here, our first experiment didn't go as well as our second experiment. This is with the light on. You can see the difference. And then this is with the lights off. The only difference this time was an instant luminol mixture that came with an oxidant, which contains hydrogen peroxide. We had a visible blue glow from the second experiment. After the experiment, we know that temperatures affects the intensity of luminol. As temperature increases, the intensity of the light also increases. We could have improved on proofreading the instructions and ingredients. And our hardest task for this experiment was measuring the luminol because it was staticky and very hard to measure. In conclusion, we learned how temperatures of water affects the glow of luminol, why the glow stops after a certain amount of time, and the importance of proofreading.
Hi, my name is Nicholas, and I had to find how much calories were in one piece of food. which was marshmallows, Cheetos, and mixed nuts. And what I had to start off with was the procedure, which I needed a ring stand and 2 rings, which, as you can see in the photos right here. And then I needed to put a skewer between like the pieces that you open the can from, and then the can has to be empty from like the soda and everything, and then you had to fill it in with 50 milligrams of water, and then you needed to put a paper clip on the piece of food that you were gonna burn, and that would have been Cheetos, mixed nuts, and marshmallows. And then once it's burning, you cannot touch anything at all. Once it's fully burned, you can start messing around with it and just like taking the piece of food off. And then I had to measure here, I had to measure it in grams, which came out to 0.0 grams for the Cheetos and the mixed nuts. And for the marshmallows, I got 5.7 grams, and then that helped me out to find how much calories were in each piece of food. So and then I had to do some math, which high schoolers helped me with, which was 31 calories for the marshmallows. Cheeto Puffs, I had 51 calories, and the mixed nuts, I had 41. And that's basically it.
Hello, my name is Sasha Nachawan. I'm from grade 8, and I made a gel electrophoresis machine. It is used for analyzing RNA and DNA and identifying someone with their DNA, or seeing if two people are related, and also finding out if someone has COVID-19. And first, my leading problem is how can someone compare different macromolecules in DNA and RNA, which is another use of the machine. I made my own machine. This is all the materials that I used. I used food dye instead of RNA and DNA because I wasn't allowed. And I ran the food dye instead of DNA across in the machine, and it did not work the first time, and it took me a long time to realize why. I realized it from one of these photos that that one of the electrodes were in the wrong place, and that is why it was wrong. And I fixed it in the prototype redesign, and I tested it once again, and it worked very well. One of the fun facts is that it was made in the 1930s, and it's still not very unpopular, which is not good because it has a lot of uses. It's very, very useful, and that's why I believe it should be very popular, and that's the reason I'm doing this project, to represent it, and so more people are knowledgeable about this. It was made by Arnie Tiselius, and one of the really great things about it is that you can see if someone has SARS-CoV-2, which is the bacteria for COVID. Basically, you run their DNA, you take their either their fingerprint, their hair piece, or anything like that, and then you mix it with an enzyme to make it a liquid, and you put it in the machine, you run it across the gel. It's negatively charged, so it runs across to the positive one, and you can see, and there's a series of unique bands that forms after you process it, and that's unique to each person. That's how you identify a specific person with it. So if there's a particular clear band at one particular place, that means that person has COVID. And I did this. My conclusion was that it works, of course, and that red dye has the smallest macromolecules, which was basically my leading problem, is how one can compare different macromolecules in DNA or RNA. I used food dye. I used red food dye, blue, and green. So the red one went the furthest. That means it has the smallest macromolecule, so it can quickly wiggle through the gel. It went 5 centimeters, so that means it has the smallest macromolecules. And blue food dye did not go that far, so that means it has the biggest macromolecules.
I am Akhil, and this is about if robots can play games or not. Like, we came up with this thinking if robots can play games or not. So this is the basic programming of rock paper scissors. Like, you can go against it. This is unbeatable tic-tac-toe. Amar, you speak about it.
Yeah, I'm Ritik. So I created this unbeatable tic-tac-toe player. It uses AI to make the best move possible, so you cannot beat it. It's unbeatable. So you can just go, and let me show you. So, yeah. I lost. So, yeah, you cannot beat it. It's unbeatable. So, we were supposed to do 3 projects, but one of it unfortunately did not work. So, do you know the Chrome Dino game in Chrome? That one, we made a project that would play the game by itself, but it did not work due to technical faults. And we created these 2 robots that plays these games with you. Yeah. Thank you.
Hi, my name is Stefania.
And my name is Sakshi.
And our project is how many cells can fit inside a piece of pencil lead?
So the purpose of this project is to find out more about cells and how many cells can fit into regular objects. We use pencil lead because it's something that everybody loves and uses.
Yeah, so I want to clarify first, what is lead/graphite? So technically there's no lead inside a pencil. It's just a term people created over the years. Graphite, however, if you're wondering, is a form of carbon that's mixed with clay to get the pencil texture. We also have research about the 4 main types of cells.
Epithelial cells,
These are cells attached to each other very tightly.
nerve cells,
These are cells specialized for communication.
muscle cells,
These are cells for movement.
and connective cells.
These are cells in the human body that help organs.
So different types of cells have different roles to play. Cells are spherical, elongated, and spindle-shaped.
So I'd like to say our hypothesis. We hypothesized that about 200,000 cells could fit into a piece of 0.5 mm lead, and about 300,000 in a piece of 0.7 mm lead.
And about 400,000 cells would fit in a 0.9 mm lead.
Yeah. So I'd like to show what materials we used.
So lead was 0.5 and 0.7 and 0.9. We used magnifying glasses, microscopes, blue stain, and also glass slides.
We used cover slips, a dropper, ruler, calculator, and pencils. So this is our procedure we did.
So to find the cylinder, we used the formula pi radius squared times height. So we did 3.14 times 0.0625 times 60 mm. 60 mm is the length of each lead that we're using, so which equals 11.775 cubed mm. Now we divide 11.775 cubed mm by 0.0001, which is each cell. So we found out that the total is 117,750, and this is the total number of cells that can fit in a 0.5 piece of lead. So we use the same formula for all the other leads, but we just use different numbers for 0.7 and 0.9.
Yeah, so in conclusion, like Sakshi said, there are 117,750 cells inside 0.5 mm lead and 230,790 cells inside 0.7 and 381,510 cells inside 0.9 mm lead. So we actually have a little experiment for you to do if you want to see your cheek cell. First, we want you to take a toothpick.
So this is a toothpick, and we're going to use it to...
Sure, I will do it. I'm going to rub it in my cheek.
So this is called a glass slide, and we're going to take some blue stain, and then we're going to place it on top, just like this.
And now I'm going to rub it.
You can really add it.
Yeah. Okay.
So now we're going to add the mini cover slips on top.
These are really thin. These are to make sure nothing gets on the microscope, and now I can see it.
So now we can see the cells.
My name is Gigi,
And my name is Mira.
and our project is basically find out if caffeine affects plant growth.
We hypothesized that if we add coffee to the
plants that it will grow taller and faster.
We discovered that adding 2 tablespoons of coffee doesn't affect plant growth.
And it would affect it negatively more. Some of our background information was that caffeine is a stimulant which increases the biological process in humans as well as plants.
Basically, some of the materials we used were a plant tray, soil, 2 tablespoons of coffee, and we used a grow cart to grow plants.
The purpose of the experiment was to determine if caffeine helps the plants grow taller or faster.
I am Yusuf Dawood. My project was about Air Force jet paper airplane. I made 3 jet paper airplane and 3 normal ones. The normal ones was tied harder than a normal one, so it went 285 and 202 inches and 285 inches. That was the first try, second try, and third try. Air Force One airplane went 124 and 133 and 356. I mean, my background information: the F-35A is the U.S. Air Force latest 5th gender fighter. It will replace the U.S. Air Force again felt off F-16 fight Falcon and A-10 Thunderbolt.
Hello, my name is Eliana and this is my partner Vanessa. Our project is on how sugar affects the strength in gelatin, and our hypothesis is the gelatin that we put 6 tablespoons of sugar in will be the strongest. Our hypothesis was supported because when the sugar and gelatin and hot water and cold water mix together, it creates like a hydrogen bond, which is an attraction to like atoms.
So basically how we kind of did it was we grabbed 9 of the cups and we had 3 trials. So then each group had, 1 cup had 2 tablespoons of sugar, 4 tablespoons of sugar, and 6 tablespoons of sugar. And basically we grabbed 100 grams of weight and we put it onto the cup, and we measured how much it went down in like about a minute. We left it for a minute and it went down by like an inch or half or so, and we found out that the 6 tablespoons of sugar was stronger and it went down the least out of all of the cups. And our purpose was that different amount of sugar affects the strength of gelatin, and another purpose is to learn about the molecular comparison of gelatin and sugar and how it's like fixed and everything.
Gelatin is typically used in foods and pharmaceutical products, and it's typically made with things like cattle hides, bones, and some types of fish skins. Sugar is typically used for baking and cooking. Another word for the sugar we used is sucrose. Sucrose is formed when temperature, like sucrocrystals are formed when temperature drops and like they bond together. Sucrose crystals, like each molecular of sugar is placed and put in forms in like a certain pattern, and it's mostly found in plants.
I think that's all we have, and like some of the materials we used were kind of just the packets of gelatin, sugar, boiling water, some cups, spoons, rulers, cutting boards, measuring cups, some gloves, and then our 100 grams of weight, and then like a tablespoon.
Hello, I'm Jaden, and this is my project. How strong is paper? So my purpose is to learn the best shape to support paper and also to learn more about paper. My hypothesis? If paper towers are made in different prisms, then the square prisms will hold the most weight. Some background information off to the side. Some older versions of paper use recycled fishing nets. The Silk Road is responsible for the mass production of paper. Where trees are chopped down is to turn them into wood and then into a pulp. There are two ways to make a pulp also. Mechanical pulping, in which the fibers of the wood are separated from the lignin, which forms wood, and chemical pulping, in which the lignin is dissolved. The ancestors of the paper are tree bark, clay tablets, and many more. Materials: 40 11 by 8.5 pieces of paper, 5 11 by 17 pieces of paper, and tape. So the procedure is take 5 11 by 17 pieces of paper, fold them into a circle, square, a rectangle, a triangle, and a pentagon. Tape them to hold them in place, and pull a piece of 11 by 8.5 paper until a paper shape follows. Here are the observation data and trials. So the average for the paper was 34 papers, or 782 grams. And the square and the pentagon were the best. The circle was the second best. The triangle was third, and the rectangle ended up in last. So the square was very steady and did not cave in. It also held the corners very well. The rectangle was not steady, caved in, and couldn't hold the corners very well. The pentagon, steady, didn't cave in. Also held corners very well. Some circle was somewhat steady, it didn't cave in, and holds those corners very well. Triangle, steady, started to cave in, but it held the corners very well. Also, the footnote here, all shapes had an issue with balancing on the paper prisms. So my conclusion was that my hypothesis, if paper towers are made into different prisms, then the square prisms will hold the most weight, was supported. And it was partially supported actually. The square did hold the most weight, which was 920 grams, to clarify. An 11 by 8.5 piece of paper is 23 grams, generally. And not only was just the square, but it was also the pentagon. The pentagon, it would make sense because a pentagon can be made out of two shapes, a square and a triangle. And the others failed for good reasons. Triangles don't have an equal perimeter like a square or pentagon, to be honest, and it generally couldn't handle the weight of all those other corners, so it couldn't make it. Circle, it's good when it works in tandem with other circles, so it could hold the center very well, but it can't handle the corners. And rectangle, well, it was good when it came to width, but in length, it was not that good. I also was going to use full 11 by 17 paper that wasn't unfolded as a control group, and also use more paper, but I decided against it. And that's my project.
Hi, my name is Eleanor. My project was, do plants grow better hydroponically or in soil? A hydroponics unit is a unit that can grow plants like this. It works with a water tank in the bottom, a tube, and the tube goes up, and it goes all the way up to the top, and then the water comes out of these spouts down to these. My materials were the hydroponics light tower, the hydroponics solution, distilled water, tap water, radish seeds, Miracle Grow brand soil, the hydroponics caps, the hydroponics sphagnum peat containers, which acts as the soil, and the hydroponics pots, which is what holds them. My hypothesis was that if plants are grown hydroponically, they will grow faster and quicker than the plants grown in soil. And the purpose of the experiment was to find out how to grow plants quickly and more efficiently, so those interested in growing plants can know the best ways to grow them. My procedure was to make holes in the soil, plant 2 to 4 radish seeds into the holes, and then cover them with soil. Then I put together the hydroponics tower and the hydroponics units. Then I cut holes into the sphagnum peat pods. And then I put the pods in the hydroponics cups. Then I mixed the distilled water with the tap water. I put water into the hydroponics tower unit. Then I monitored over the next few months. My hypothesis that the hydroponically grown plants would grow faster was initially supported, but could only be partially supported in the end due to the hydroponically grown plants dying. I would improve that by adding more solution, because I think the reason why they died is due to not having enough vitamins compared to the plants grown in soil. This mistake was most likely caused by my own action, so that is what I would improve. Overall, my hypothesis was partially supported, and could it be improved in the future?
My name is Carissa.
And I'm Gabriel.
And our project is about, is it possible for a water balloon to erupt indoor flame without erupting? So, you talk. The balloon did not pop. The balloon did not pop, so the purpose of a water balloon is to place under a flame that it will not pop. The hypothesis is the purpose of this experiment is to see if a water balloon can fight fire and not pop. Another purpose is to learn about the property of water, such as specific heat. You talk.
Say the background.
Background information. Balloons are made of rubber, latex. I don't know if it's. Plastic or fabric. When an air filled with balloon is explode to fire, it will pop. It will burst because air is a pore of heat. Water is a compound made of hydrogen and oxygen. It has a high heat capacity, which means it takes off energy for it to heat up.
And make sure you are in a safe location. Make sure hair is tied back and all strings on clothing are out of the way. Using a light a candle, blow up the balloon and tie it off. Place it over the candle and record all observations. Repeat steps 5 and 6 more times. Place 60 milliliters of water in a balloon. Place it over a candle and record all observations from your senses as possible. Sight, sound, smell. Repeat 8 to 10 more times.
You say it.
Oh my gosh. The hypothesis for this experiment is if a water balloon is placed under a flame, then it will not pop due to the water absorbing the heat. This hypothesis was supported. According to the data, each balloon that was filled with the water did not pop. Within a few seconds, as expected, on all balloons there was a black soup type stain due to the flame. The water balloon did not take long time for it to heat up as well as cool down. If this mild day was no wind, the result would have been better if the candle did not blow out a few times.
It was very windy outside, so the candle kept going out. The second balloon that had water kept blowing out the candle. The wind kept blowing out the candle. There was 2 balloons with water and 2 without. The first balloon we put over the candle was without water. When we did the first balloon without water, it did not immediately pop. when we put the balloon closer to the fire, it popped. Before moving the balloon closer to the fire, the balloon started to shrink. The balloon without water took six seconds to pop. The balloon that had water did not pop at all. The more you hold the balloon over the fire, the more black burn marks are at the bottom of the balloon. The balloon without water did not pop because the water absorbed the heat from the flame.
My name is Aya. My name is Symphony. And this is our STEM Fair project. Which vegetable can make the best dye? So for the first purpose for our experiment is to know which vegetable can make the darkest dye. The second purpose is to find out the chemical makeup of the vegetables and which chemicals cause their color. Background information. Beets are a root vegetable that is a rich source of a group of red and yellow pigments known as betaines, comprising red-violet betaxines and yellow betaxantins. The rosy betalin-rich juice of red beets was used as a cheek and lip stain by women during the 19th century. Red cabbage leaves a substance called anthocyanins. Capsaicins makes fully mature bell peppers red. Chlorophyll molecules absorb light energy in the blue. Hypothesis. If vegetables are used as dyes, then the beets will have the darkest stain. Independent variable. The dependent variable is the same color. Constant. Same type of fabric, same time within the liquids. Control group. The fabric not dyed.
The materials used during this experiment were 2 white cotton t-shirts, vegetables which were beets, red bell peppers, red cabbage, and spinach. 4 rubber-made containers, 4 pots filled with water, cutting board, knife, tongs, gloves, and paper towels. The procedure to make this experiment was to make the vegetable liquid, cut each vegetable into quarters. Place each vegetable in a separate pan and boil the vegetable for 20 minutes. Allow the liquid to cool. When the liquid cools, remove the vegetable from the pan and discard the vegetables. Place the cooled liquids in the separate containers and label them. Cut cotton t-shirts into 12 pieces. Place 3 cotton swatches into each container and seal it. Wait 24 hours and remove the swatches for observations and record all observations. The observations and data that we have are: the beets made the darkest dye for the swatches. The color resembles a red purplish dye. The red cabbage made the strongest smell for the swatches. It smelled the worst out of all the vegetables and made a purple colored dye. The spinach did very little in color to the swatches. It was a light green color. The red bell peppers made the swatches barely look dyed. It was the weakest. The liquids were different too. The beets and cabbage liquid were opaque and the spinach and red bell pepper liquids were transparent. In conclusion, the hypothesis is that if a white cotton t-shirt swatches are placed into different vegetable liquids, then the beet liquid will cause the darkest stain. The hypothesis is supported. The experimental data shows that all 3 cotton swatches were heavily stained with beet juice. Beets made the darkest dye for our swatches. This makes sense because it was used as a makeup for lips and cheeks in 1800s. The chemical in beets that caused the stain is biotin. While red cabbage made the most vibrant scent, red bell peppers produced the clearest dye due to its water composition. Spinach, however, made the lightest dye, possibly due to its water composition. The experiment can be improved by soaking the swatches for a longer period of time.
And that's it.